name is Emme Baldwin, and my study is on the relationship between CPAP compliance, prescription medication compliance, BMI, and other demographic factors. In the top left corner of the poster, you will find the introduction and purpose to my study. The purpose of the study is to see if a relationship exists between compliance with CPAP and prescription medications and OSA patients. The study will also look at BMI, age, and gender, and see if a relationship exists between those factors and CPAP compliance. If a relationship exists, it could be helpful for providers to determine which patients need extra attention and interactions when giving out medications and treatments. This could help keep more people compliant. Underneath this section, you will find the review of literature. So at this point, you might be wondering, what is OSA? OSA stands for obstructive sleep apnea and is defined as frequent periods of apneas and hypopneas. Apnea means temporary stoppage of breathing. Hypopnea means slow or shallow breathing. So when you put it together, OSA is a mix of temporary stopping your breathing and slow and shallow breathing. OSA is treated with continuous positive airway pressure, also known as CPAP. OSA can negatively impact people's lives in many ways. There are many health impacts of untreated OSA, like increasing risk of high blood pressure, stroke, etc. CPAP compliance. CPAP is the main treatment for OSA. The main reason for untreated OSA is lack of compliance with CPAP. There are many factors that predict a patient's compliance with CPAP, like the severity of OSA. Medication compliance is the likeliness of a patient to follow a doctor's advice when it comes to a medication. Demographic factors. I read about how CPAP compliance affects BMI, and I wanted to dig deeper into this relationship and learn more about it. The chart below shows how I believe each factor will affect each other. I think that CPAP compliance affects medication compliance, and I think that demographic factors may affect CPAP compliance. This chart now leads me to my hypothesis, which is found in the section right below the chart. My first hypothesis is people with poorly treated OSA are more likely to be non-compliant with prescription medications. My second hypothesis is people with a higher BMI are most likely to be non-compliant with CPAP. My third is demographic factors like age, gender, impact a person's compliance with CPAP. In the top middle section, you will find my methodology. I received blinded data from a healthcare company's pharmacy. In this data, I received 49 patients with OSA. 12 of those patients were compliant with CPAP. 12 of those were non-compliant with CPAP, 31 were compliant with CPAP, and 6 were uncertain. The data includes age, gender, BMI, CPAP compliance, number of prescription medications prescribed, amount of prescription medications compliant. What are the compliance definitions? Medication compliance will be defined as fulfilling the medication at least 75% of the time. CPAP compliance will be defined as using CPAP for at least 4 hours per night and for 70% of the nights. In the section below this, you will find the results. You can see all 49 patients' data, all of it is blinded. You can also see a few examples of the graphs that I will be using and entering the data into. In the top right-hand corner, you will find the discussion, and in the discussion you will see my timeline. In February of 2020, I received the pharmacy data. In March of 2020, I reviewed the data. In April, I began analyzing the data, and I'm still doing that now. June 4th is the Science Research Symposium. In the summer of 2020, I will try to get more data if I deem it necessary, and I will work on analyzing the new and the old data. Senior year, I will present at competitions. Underneath this section, you will find the conclusion. This study is looking to see if a relationship exists between CPAP compliance and prescription medication compliance. I'm also looking at the data to see if there is a relationship between BMI, age, gender, and CPAP compliance. All of this is done using blinded pharmacy data. If my hypothesis is proven correct, Then, providers will know when prescribing prescription medications to non-compliant CPAP users, they will need to give extra attention to keep those patients compliant with other medications. Providers will also know if they need to pay more attention to CPAP compliance for patients with specific demographic factors. Underneath the conclusion, you will find the references, all of the sources that I cited to make this PowerPoint. Thank you.
My name is Katherine Bergan, and my research question is on hypertonic skin cells as an early symptom of pancreatic ductal adenocarcinoma. Pancreatic ductal adenocarcinoma, or PDAC, is the most common type of pancreatic cancer. This disease begins in the pancreatic duct, but usually it's not detected until the tumor spreads to other organs. Because of this, the survival rate is extremely low. Obvious symptoms such as jaundice and abdominal pain only occur after metastasis, and there are no known and reliable early symptoms to catch PDAC early. PDAC obstructs the pancreatic duct, so bile is directly affected as it is supposed to flow to the duodenum. Instead, bile salts will enter the bloodstream. Bile salts in the bloodstream will be close to the surface of skin, and surrounding cells will become hypertonic in the new environment. My hypothesis is that patients with PDAC will experience dry skin in the early stages of their illness since skin cells have lost water. I've recently contacted the Upstate University Hospital for data that I'd like to use as an analysis to prove a strong positive correlation between PDAC and dry skin. I'm currently looking through what they sent for a good data set. I've also contacted Skidmore College for a space to conduct a 3-trial experiment in which bile cell is put on skin cells and the reaction is recorded. For the rest of this month, I'd like to finish and submit my research plan. In a few weeks, I'd like to be done with my data analysis and have set a date in the summer to conduct my experiment. In the fall, I'd like to start thinking about my final paper. I have not yet collected results, but I am expecting the data analysis to prove a correlation. I'm also expecting each trial of my lab experiment to show that the cytoplasm shrinks when in contact with bile salts. A supportive data analysis would prove real-life impact, which paired with the microscopic backing of a lab experiment would help build understanding of PDAC in the medical community. With knowledge of an early symptom of this deadly disease comes a diagnosis before the tumor grows beyond control. Patients will have a longer life expectancy, allowing more time for testing and treatment that will increase the survival rate of PDAC.
Hi, my name is Cristina DeMeo, and I will be studying the effect of various small molecules on the disease progression of amyotrophic lateral sclerosis. Amyotrophic lateral sclerosis, or ALS, is a neurodegenerative disease, and it causes the death of both the upper and lower motor neurons in the brain. After slow paralysis of the extremities, patients with ALS experience death within 3 to 5 years of symptom onset. ALS can be caused by one of several gene mutations, most commonly a mutation of C9orf72, or chromosome 9 open reading frame 72. On a cellular level, symptoms of ALS include excitotoxicity, which is the imbalance of neurotransmitters, increased free radicals, which lead to oxidative stress, RNA foci, which are toxic RNA, and endoplasmic reticulum stress, which causes proteins to be misfolded and leads to aggregation. One of the most successful tools to help patients with ALS has been the use of small molecules. Small molecules have a low molecular weight, and they are organic compounds that regulate specific cellular processes. Small molecules have seen a recent growth in popularity because they are small, they are cheap, and they can be taken orally, which means that they are ideal for experimentation. There are thousands of small molecules and thousands that haven't been tested. Often those that are tested on animals. And although this is a valuable model, human-derived primary cells are an extremely valuable model because they react very similar to how a human would react. Since small molecules can target and regulate specific cellular processes, and since primary cells are such a valuable model for experimentation, if C9orf72 mutated human-derived primary cell lines are exposed to various small molecules targeting a specific symptom of ALS, then the overall disease progression of ALS will decrease. I will obtain human neuron-derived primary cell lines from my advisor. They will be cultured in standard culture medium and supplemented with standard antibodies and standard growth serum. There will be 2,000 cells and 50 microliters of medium per well, and they will be cultured for 24 hours. The cultures will be stained for better visibility. After 24 hours, the cells will be broken up into two different groups. One group will act as a control group and will not receive any exposure to small molecules, and the other group will be exposed individually to 20 different small molecules. The small molecules will be added to a final concentration of 10 micromolar, and each molecule will be tested twice in an attempt to decrease experimental error. The cells will be tested 24 hours after small molecule introduction to determine disease progression. Some of the small molecules I'll be using will be targeting inflammation. Others will be regulating RNA, decreasing RNA foci. Some will increase autophagy, which is a cellular process that regulates protein aggregation. Others will decrease endoplasmic reticulum stress. And finally, some small molecules will balance glutamate and GABA, neurotransmitters in the brain, and decrease excitotoxicity. So after the 24 hours, the disease progression will be determined by 3 different tests. Both groups, the control group and the experimental group, will be tested, and statistical significance will be determined using an ANOVA test and bioinformatics analysis. One of the tests I will be using is the RNA FISH test, or RNA fluorescence in situ hybridization test. This test will be able to identify RNA foci. The test uses fluorescent probes which will bind to specific nucleotides, and I will be able to determine how many RNA foci in the cell. An increased amount of RNA foci will mean an increased disease progression. The next test I use is the Western blot, and similarly to the RNA FISH test, this test uses antibodies that will bind to specific proteins. Increased protein aggregation will mean increased disease progression. Finally, I will use the hemocytometer tests, which will be able to determine neuronal death. This test includes a small grid that can be placed under a microscope, and I will be able to count the cells. Decreased cell count will mean increased neuronal death and increased disease progression. Over the next few months, I will be finalizing my research plan, completing any paperwork necessary for my experiment, and in general, getting ready for the experiment. In summer or fall, I will be able to conduct my experiment and begin any statistical analysis. So in conclusion, I will have 2 groups of human neuron-derived primary cell lines. Both groups will have a C9 or 72 ALS-causing mutation. One group will be exposed to 20 different small molecules, and the other group will not have any exposure. There will be several tests used to compare these 2 groups, and these tests include the RNA FISH test, the Western blot, and the hemocytometer test. Thank you.
Hi, my name is Ava Herzog, and I made a study called The Effect of the Retinal Degeneration Mutation RU1 on Circadian Locomotor Activity and Nesting Behavior in Mice. So circadian clocks are networks of genes and protein that control our everyday rhythms such as sleep. Running wheel activity is a common way used to measure the output of the master circadian clock known as the CSN, which regulates these circadian rhythms. Nesting behavior is commonly used to quantify the general well-being of mice. The retina is involved with light detection and visual recognition, receiving light signals through the photopigmental opsin, a light-sensitive retinal protein, which embeds retinal neurons known as the iPRDCs. It then is able to convert these into neural signals that the brain can understand. Another important part of the retina are the photoreceptors. The rods are involved in mediating dim light vision, and the cones are the photoreceptors involved in mediating bright light vision. These photoreceptors are not technically needed for the entrainment or synchronization of the biological light, but have been shown in previous research to provide some sort of input to the clock. The loss of these photoreceptors is known as retinal degeneration, causing visual blindness. Retinal degeneration is due to the homozygous recessive RU1 mutation. Retinal degeneration does not affect the retinal neurons that I mentioned previously. This same mutation causes autosomal recessive retinitis pigmentosa in humans, having the same impaired retina. So from this, I decided to determine whether or not the RU1 mutation affects running wheel activity and nesting behavior in mice, more specifically if the RD1 genotypes, gender, or differences in light intensity affect this. So for my study, I was able to get 56 mice, 28 males and 28 females. They were of the F2 generation, descended from the C57BL6J, a control strain, and the SJL strain, which is homozygous for the mutation. So I would hypothetically have the perfect Mendelian ratio, having 25% being wild-type mice, as shown in the purple, 25% being the blind mutants, as shown in the orange, and 50% being carriers, as shown in the green, having half the number of photoreceptors and being partially blind. So my study had 3 main behavioral observational periods. The first 10-day period measured the wheel activity in a 12-hour light and 12-hour dark cycle. The next 10 days measured the wheel activity in total darkness. And the last 5 days measured the nesting in the same 12-hour light and 12-hour dark cycle. For the 2 12-hour light and 12-hour dark cycle, half the mice were in a bright light room and half the mice were in a dim light room, so I could look at the effect of varying light intensities. I quantified nesting by calculating the difference in initial and final cotton weight and then dividing this number by the animal's body weights, which I took at the beginning of my study. This is to account for the fact that bigger mice tend to build bigger nests. Each running wheel had a microswitch, so I was able to get automatic data collection through the VitalView program. I measured activity as revolutions per every 10 minutes. I then used the ActiveRhythm software in order to create actograms, which show activity over time, and periodograms, which are used to calculate the length of the circadian period. I then reached out to the Transnet Wax Genotyping Company in order to determine the genotypes. I used the SCS system for analysis of variance. In the left graph, you can see that nesting behavior was affected by the interaction between genotypes and genders, with the female and male carriers having drastically different activity levels. The right three graphs illustrate the direct effect of the mutation on the running wheel activity, with the carriers, the partially blind animals, being the most inhibited. These results show the effect of the heterozygous RD1 mutation on both animal well-being and local motor activity. Some other significant results from my study were the more active females and the generally greater activity in dim light rather than bright light. The stability of entrainment as measured by the circadian period's length in the 12-hour light and 12-hour dark cycle was significantly affected by an interaction between dim light and the mutants and the carriers, but all of the mice were able to entrain, so this small but significant result is better representative of the slight activity level differences rather than their ability to entrain as a whole. The free running period, which is the length of the circadian period when in the total darkness period, was not affected by the mutation but was found to be greater in males. From this, I concluded that the RD1 mutation directly affects running wheel activity and nesting behavior in mice. It is not through the biological clock, since the free running period, which is the length of the circadian period in total darkness, was not affected by the RD1 mutation, was only significantly changed by a sex difference. The carriers, those being partially blind and having half the number of rods and cones, were the most affected, showing that partial blindness may be more detrimental than complete blindness. For future research, I would like to examine this direct relationship and the possible extrapolation to humans.
Hi everybody, my name is Jorgensen and I am presenting my poster on the effect of human social distancing due to the 2020 COVID-19 outbreak on the behavior of pet dogs as assessed by the CBARC. So essentially, my study is focused around the fact that behavior is molded by environment, and a lot of humans' environments are changing quite rapidly right now due to the pandemic that we are experiencing. And the goal of my study is to analyze how the pandemic and the humans' changed behavior is affecting dogs and how it's going to impact dog behavior. So if you think about it, if you're staying home extended periods of time during the day, how is that going to affect your pet dog? That is essentially what I'm trying to figure out. My hypothesis is that increased time spent with the dog's owner will lead to behavioral changes both when the dog is interacting with other dogs and when the dog is interacting with humans. Just because their daily life has changed so much and their behavior is likely to change in response to that. So I used a Microsoft form to have participants sign up. I had 70 participants sign up originally. I gathered my participants through Facebook. My mother was part of an academic group, and so she sent out the link to it, and the link was spread throughout Facebook with those participants continuing to spread the links. I had 70 participants sign up originally, and the study was 6 weeks long. So at the very beginning, I had those 70 participants fill out some consent forms and things of that nature, but I also had them fill out the CBARQ, which is the Canine Behavioral and Research Questionnaire. Essentially, the CBARQ analyzes a dog's behavior and points out any areas where the dog may be having some issues in their behaviors or where there are some areas that the dog is significantly well behaved. And this can kind of help owners and as well as other dog trainers figure out what the problem areas are in a dog's behavior and what kinds of things need to be addressed. Then after they take the CBARQ, a 6-week stretch of time is going to elapse. Essentially, every week I will be sending out a survey just to find out, what's been going on in the dog's life, to make sure that there's nothing changing other than the human's quarantine that could skew the dog's behavior. And then at the end of those 6 weeks, the dogs will be taking, the humans will be taking a CBARQ again to analyze the dog's behavior. And I can then take those two CBARQ scores and compare them to analyze if there's been any change in behavior over the 6-week time period in which the humans were social distancing and quarantining. So that can be analyzed to determine if there is a change in behavior. No conclusions have been formed to this date. However, I'm continuing to work on my study. As of right now, I'm working towards my statistical analysis portion. So yeah, thank you.
I'm studying the effect of selective breeding on tolerance to drought in Wisconsin fast plants. Drought has been a growing concern in the scientific community since the early 2000s. Drought can be defined as a period of below normal precipitation that limits the plant's productivity. Drought can cause a disruption in homeostasis to the plant as well as decrease the plant's water potential. Therefore, scientists have been starting to look for ways to increase plants' stress tolerance to drought. The most common type of experiment has been scientists modifying the plant's genes within a laboratory setting. However, this can cause many shortcomings and therefore skew the data. Through my research, I have found that the method of selectively breeding the plant to increase its stress tolerance is not something that has been commonly used. Therefore, in my study, I will be selectively breeding the Wisconsin fast plant to see if I am able to increase its stress tolerance to drought. My hypothesis for my experiment is that if Wisconsin fast plants are introduced to a drought stressor and replanted by selective breeding, then after several generations, the plant's tolerance to drought will increase. Through this study, I am hoping to decrease the need for harmful chemicals in the environment and provide a more environmental friendly way to help the plants grow and flourish. To conduct my experiment, I will be splitting the plants up into a control group and an experimental group. For 2 weeks, both the control group and experimental group will grow under normal conditions. And then after that, the experimental group's water supply will be cut in half and it will experience drought for about a week or a week and a half. After that, I will uproot both the control group and the experimental group, and I will cut the plants where the stem meets the root. I will then measure the mass of the root and the mass of the stem. After that, I will look at the root mass relative to the stem mass that it is supporting. After these measurements and comparison have been taken, the healthiest plants of each generation will have its seeds taken out and replanted. Before each generation is replanted, I will take a couple of seeds from each generation and set them aside till the end. And after that, I will repeat the process for several generations. After the last generation has been planted and the data has been collected, the seeds that I have gathered from all of the generations will then be planted. That way I can get a clear picture to see if the drought stress tolerance has increased from the first generation to the last. While a statistical analysis test has not yet been decided, I have figured out several charts and graphs that will help to organize my data. The first chart will help me organize the average root mass, average root stem, and the ratios of each generation. The bar graph below will help me compare the control group and the experimental group of each generation and help me to get a clear picture to see if the experimental group's drought stress tolerance has increased from generation to generation. I have been fortunate enough that this global pandemic has not thrown off my plan to start growing by too much. So I have found that I will be able to submit my research plan and begin my growing after gathering my materials by June. Then from there, the rest of my summer will be used to plant the generations and gather my data. By the end of August, I am hoping to have all of my data completed. That way, when school starts in September, I can finish analyzing my data and sorting it neatly into data tables, and I will be able to start working on my presentation to be able to compete. In conclusion, this study is to see if through selective breeding, I am able to increase the Wisconsin fast plant's stress tolerance to drought in a natural way without needing harmful chemicals. Thank you.
Hello, my name is Isabella Rando, and I am testing the effects of this antioxidant supplement acai on oxidative stress levels in Drosophila melanogaster with superoxide dismutase mutations. I came up with the idea for my research after reading numerous articles about oxidative stress, which is when the body contains a large amount of free radicals and a small amount of antioxidants. I also read about the negative effects that oxidative stress has on health and lifespan. I learned that certain mutations, including the mutation that I decided to focus on for my research, superoxide dismutase mutations, can lead to the formation of free radicals, which in turn cause oxidative stress and shortens lifespan. This is because superoxide dismutase is a biological antioxidant defense mechanism, and antioxidants are essential in preventing oxidative stress. So when superoxide dismutase is not functioning properly, we become more susceptible to oxidative stress. Previous research has shown that acai can reduce oxidative stress caused by environmental factors, including hydrogen peroxide and paraquat, and blueberries can upregulate superoxide dismutase. However, I hadn't come across any studies that observed whether acai could reduce genetically caused oxidative stress by potentially upregulating superoxide dismutase. So I am aiming to determine whether acai, which is an antioxidant, can extend lifespan in flies with a superoxide dismutase mutation, or a SOD mutation. I have hypothesized that if Drosophila melanogaster with superoxide dismutase mutations consume antioxidant supplements, particularly acai, then they will have a longer lifespan when compared to Drosophila melanogaster with superoxide dismutase mutations that do not consume acai. To test this hypothesis, I am going to observe 4 groups of Drosophila, which are fruit flies, consisting of about 175 male and 175 female fruit flies. 2 of the 4 groups will consist of Oregon R wild-type flies that do not have a superoxide dismutase mutation. The other 2 groups will consist of SOD N108 mutant flies, which is a mutation that is predicted to cause oxidative stress. I am going to divide those 2 separate groups of non-mutant and mutant flies even further by feeding one of the 2 non-mutant groups and one of the 2 mutant groups a sucrose food mixture supplemented with acai, which is expected to prolong the lifespan of the flies. All other variables, including light exposure and temperature, will remain constant, so that the only things that will impact the lifespan of the flies are their mutations and their antioxidant consumption. The data table and graph that I have shown here will compare the average lifespan of the flies in each of the 4 groups that I am testing. This will allow me to determine whether acai was effective in extending lifespan as predicted. I plan to conduct my research in late August and September at Skidmore College. This will allow me the appropriate amount of time required to analyze my data and draw valid conclusions, which I plan to have completed by the end of October. If my hypothesis is supported, and acai extends lifespan of superoxide dismutase mutants, this experiment could potentially be replicated using human participants, and antioxidants could be used as an easily accessible, non-invasive treatment option for individuals who are genetically predisposed to oxidative stress and who are at risk of the numerous health issues associated with oxidative stress.
Good evening. My name is Lily and I'm a junior. The title of my study is the relationship between handedness for communicative gestures and atypical lateralization for language in left-handed humans. In Homo sapiens, the brain's major structures for language processing are located in the left hemisphere. The term for describing this phenomenon, where one side of the brain over the other controls language functioning, is language lateralization. Researchers have observed a clear link between language lateralization and handedness for manipulative tasks. Handedness refers to a person's dominant hand, and manipulative tasks include activities like writing with a pencil or pen. Since the left hemisphere of the brain communicates with the right side of the body, it makes sense that most people are right-handed writers and most people are left hemisphere lateralized for language. Based on this logic, we would also expect that the opposite is true. It would follow that most left-handed writers should be right hemisphere dominant for language, but this isn't the case. Only about 30% of left-handers are lateralized this way. The other 70% are left hemisphere dominant, just like their right-handed counterparts. One way to test for lateralization is by using a visual field paradigm, in which a participant completes a language task using only one visual field, and thus only one hemisphere of their brain. A lexical decision task is a language task in which participants are presented with letter strings and asked to determine whether each stimulus is a word or a non-word. Communicative gestures are hand motions used to convey a message, and some evidence has been found in chimpanzees that unlike handedness for manipulative tasks, handedness for communicative gestures may be an accurate predictor of language lateralization. It's important to keep in mind that while around 90% of the human population is right-handed for writing, a slightly higher percentage of people are left hemisphere lateralized for language. This means that the best communicative gesture for this research is one that produces more than 90% right-handed responses. Different types of communicative gestures produce different ratios of right to left-handed responses. Based on all of this, my hypothesis is that left-handed humans who are left-hand dominant for communicative gestures are more likely to be atypically or right hemisphere lateralized for language. My participants will be around 40 introduction to psychology students at Skidmore College between the ages of 18 and 25. They will all need to be monolingual English speakers and left-handed writers. I will conduct the Edinburgh Handedness Inventory, a short survey to determine handedness for manipulative tasks, with all participants to confirm that they are left-handed. I'll then run a deictic or pointing gesture task to determine hand preference for communicative gestures. The third test I'll conduct will be a visual field paradigm lexical decision task to determine language lateralization. To analyze my data, I'll first score each participant's EHI responses and discard data from any participant who does not score as left-handed. Next, I'll use the results of the communicative gesture task to create a handedness index score for each participant. The EHI score will tell me which hand is preferred and to what degree the preferred hand is dominant over the other. PsychoPy 2.0, the software I'll use to conduct my lexical decision task, will compile time and accuracy results for each participant. All data from inaccurate responses will be rejected, and I'll take averages of response times for words and non-words in each visual field. Participants who respond more quickly when stimuli are shown in the left visual field will be right hemisphere lateralized for language. I'll run an analysis of variance with the deictic gesture task HI scores and lexical decision task results. If there is a significant interaction, my hypothesis will be supported. I have not yet begun collecting data, so I don't yet have results, but this bar graph is one of the charts I'll use to display my data once collected. It compares hand dominance for the didactic gesture task to language lateralization. My next step to move forward with my research plan is to submit my study to Skidmore College's IRB for approval. I'll also submit my research plan to ISEF for approval and begin collecting data when Skidmore students return to campus in the fall. In the late fall, through the end of my first semester, I'll analyze my data and prepare for competitions. The significance of this study stems from the fact that we don't know why a minority of left and right-handed people are right hemisphere dominant for language. If successful, this research may shed some light on this question. My study also has potential applications in early childhood education and could introduce testing for hand preference for communicative gestures as a new way to predict language lateralization. To conclude, my hypothesis is that left-handed humans who are left-hand dominant for communicative gestures are more likely to be atypically or right hemisphere lateralized for language. With my participants, who will be left-handed Intro to Psych students at Skidmore College, I'll conduct the Edinburgh Handedness Inventory, a didactic gesture task, and a visual field paradigm lexical decision task. I'll create a handedness index score with the results of the communicative gesture task and take an average correct response time for each visual field from the lexical decision task and run ANOVA to see if my hypothesis is supported. Thank you.
My name is Vania and I'm presenting my plan for my research studying the influence of outgroup and in-group race embodiment on eye movements and gaze patterns. This will be an immersive study, meaning that participants will experience the experiment virtually. So in the past few years, research on the associations between human behavior and immersive virtual environments has significantly increased. Whether for purposes as spreading awareness about a certain social or environmental issue or disorder therapy. In the majority of racial bias intervention research, the implicit association test has been used as a measure of implicit bias. However, it can be unreliable because it is solely based on reaction time. Many studies have shown that own race faces are recognized differently than other race faces. So my hypothesis is that eye movement fixations on outgroup characters will be shorter in condition 1, where the participant will be embodying a different raced avatar as compared to condition 2, where they are embodying the same raced avatar. I will have 10 to 30 female participants of similar racial background, the gender choice made just for consistency purposes. They will be between grades 9 to 12 from Saratoga Springs High School and will be conducted in the VR room. The materials that would be necessary would be a head-mounted device, or HMD, to ensure a fully immersive experience, and participants will use a Viv controller that will be strapped to their right hand to control movements in the virtual environment. Eye tracking goggle system will be used. So first, they will be assigned to 2 conditions where they either embody a white avatar or a black avatar. And they will have some time to familiarize themselves with the controls on the Viv controller and with the virtual environment. Then there will be an embodiment phase, about 5 minutes, in order to get them feeling like they are in that environment. After the embodiment phase, they will be approached by 20 virtual human female avatars, half white race and half black race. And these avatars will walk past the participants either by the right or from their left. And prior to being in the virtual experience, they will be instructed to attempt to memorize these avatar faces. And the eye tracking goggle system will be used to record the eye movements and gaze fixation points. And this process should take about 5 to 10 minutes. After the virtual experience, they will have a demographic questionnaire as well as a post-experience questionnaire. The purpose of the post-experience questionnaire is to determine the effectivity of embodiment in the VR. Number of fixations and their durations on avatar faces will be recorded, and their mean P scores will be calculated. If there is a disparity between the durations of fixations in different embodiment conditions, this may show that racial prejudice can selectively bias eye movements in a virtual space. Observing the effects of other race embodiment on attention and eye movements might be able to lead to new insights into a more effective measure of explicit racial bias.
Hi, my name is Ellie, Sweeney, Benzon, and today I'll be presenting my poster on my study on the effect of summarizing passages on an individual's handwriting following a reading of said passage within a Spanish as a foreign language context. I came to create this study by reviewing many other studies and discovering a hole within the reading comprehension, summarizing, and handwriting niche. Although the relationship between reading and writing, as well as the relationship between summarizing and reading, have both been proven within a foreign language context, the relationship between handwriting and summarization has yet to be explored, and this is what I built my study on. Thus, the aim of my study is to determine the validity of the relationship between summarizing after reading a passage and the resulting handwriting legibility in a foreign language setting. I have based my study off two hypotheses. If an individual summarizes text whilst or post reading a passage in a language other than their native language, the individual's handwriting legibility in that language will improve through means of completing summaries. My other hypothesis is that students who self-report a higher HLS score throughout the study will have a greater increase in their HLS score from pre-study to post-study. My ideal participant throughout this study is someone who is not a Spanish native speaker and someone who does not have a handwriting difficulty. If someone has a withstanding issue with handwriting difficulty, they will not be excluded from the study, but their data will be taken into account in order to ensure accuracy in the overall results. My ideal participant group is all one-level language learning class. Although there will be different levels of proficiency within this class, this will be taken into account and can be expected. There will also be different professors within each class. The materials I need for my study are reading passages in Spanish, reading comprehension activities, summary prompts for the experimental group, the handwriting legibility scale that is based on 5 categories: global legibility, effort required to read, and layout on the page, letter formation, and alterations. Throughout the study, there will be 3 groups: experimental group, control group 1, and control group 2. All participants will complete a personal questionnaire, baseline handwriting sample in English and Spanish, and self-reported HLS scores. Experimental group will complete 3 summary tasks, and the control group will complete 3 reading comprehension tasks. At week 0, all participants will complete a baseline handwriting sample in both English and Spanish. Week 1, participants will read a passage in Spanish. Experimental group will complete summary tasks. Control group 1 will complete multiple choice activities, and control group 2 will complete reading comprehension activities. This will be repeated for week 2, and week 3. After the study, all participants will be asked to complete a final handwriting sample. The HLS scores will be scored by outside raters to ensure inter-rater reliability. The results will be displayed in 2 tables, one for self-reported HLS scores and one for average HLS scores at all instances of the study. Week 0, English and Spanish, week 1, week 2, and week 3. It will also be displayed in a graph that has each instant of the study and control group on the x-axis, and on the y-axis there are the HLS scores 0 through 20, which assigns 4 points for each of the 5 categories of the HLS scale. The limitations of my study are that since it's a small sample size, there will be relatively little statistical potential of the results, and this also may limit the number of significant effects. In addition, while the focus of the study was narrow and limited to ensure control, this makes it difficult to generalize the results of the study to greater outcomes. The study will allow the relationship between summary writing and writing to be proven or disproven while simultaneously extending the relationship beyond native languages and into foreign languages. The significance of my study is that it will enable both students and teachers in foreign language and conventional classrooms to apply a summarization technique in the classroom, not only to improve reading comprehension, but to simultaneously improve handwriting within the said language. Teachers will be able to implement this vast study and useful technique throughout their classes as an efficient study method to improve two common skills simultaneously, handwriting and reading comprehension. The significance of the study will be global due to the fact the results in the Spanish language should be indicative of results in any other language. The research will also have a profound social impact in that it is inclusive to all genders, races, and ethnicities, so it will be able to be utilized across the country and across the globe due to the diversified participant. Thank you so much for watching.